Good afternoon and hello everybody. Thank you for joining to this seminar after the summer break. It's my pleasure to introduce to the new speaker that we have for the talk today, that is the Dr. Miguel Martín-Acebes. Dr. Martín-Acebes is principal investigator in INEA, in the one moment. En el Instituto Nacional de Investigación y Tecnología Agraria y Alimentaria de Madrid. He's principal investigator also in this institution from 2015. He has been working for a long time in Flavivirus, particularly in West Nile, but also in other Flavivirus like as Usutu and Zika. And the title of his talk is Metabolic Reprogramming During West Nile Virus Infection. So, Dr. Martín-Acebes, please, the floor is yours. And again, thank you very much for accepting to participate in these seminars. So go ahead, please.
So thank you, Jorge, for your nice introduction. I'm going to share my screen. So, can you see the presentation?
Yes, we can see your presentation in the whole screen.
Okay, so thank you very much again, Jorge, for your nice introduction. And today I'm going to talk to you about some part of the work that is currently going on in our lab. What is the study of the metabolic reprogramming during West Nile virus infection. So West Nile virus probably doesn't need presentation. Currently it's an important emerging pathogen, and as all of you know, this virus is maintained in nature in an exotic cycle between birds that act as natural reservoirs and that can jump to humans or to other accidental hosts through the bites of infected mosquitoes. Well, if you are a horse, you are lucky. There are very good horse vaccines. They elicit very good protection, and you vaccinate the horse and you can forget about West Nile virus. But if you are a human, you are not so lucky. There is no vaccine for humans, so you can get infected by the virus and develop a severe illness. And this illness is called West Nile virus disease. And in the most of cases, it is asymptomatic. In 20%, 25% of the cases develop what is known as West Nile fever. That is a mild flu-like syndrome. And in less than 1% of the infections, this leads to neuroinvasive disease that can be fatal and cures with encephalitis, meningitis, or acute flaccid paralysis. And if it is not lethal, it can induce neurological sequelae that can be long-lasting. So this is the top of the pyramid, the cases that we are going or we want to treat. Well, as I told you, is you get infected with West Nile virus and you became ill. There is no antiviral therapy approved, and the treatment is only supportive. It includes antibiotics, hydration, and in some cases, treatment with anti-inflammatory like corticosteroids. So we are interested in the lab in the develop or new antivirals, identification of new antiviral targets, and try to find some therapeutic opportunities to fight this virus. And when you are going to plan to design antivirals, you can target the two aspects of the infection. You can target the virus. Here you can see on the right. This is the classical approach. The compound, the drug, is directed against a viral factor that it is very different from cellular factors. So these antivirals are very specific, and usually they are directed against one specific virus. On the other hand, you can treat the host. In this case, you can act on a cellular factor that is required for viral multiplication. And when you block this cellular factor, well, you block viral multiplication. And this has a potential advantage, that is that the host factors that are shared between different viruses are prone to be the candidates to develop broad spectrum antivirals, which can be useful for emergencies, for novel pathogens, or virus highly related to your target. So here you have a summary of the pros and cons of the virus targeting antiviral approaches, which the pros are the selectivity, because they go against viral targets. This is reinforced because there are very successful drugs already in the market, and the procedures for efficacy are clearly established. The cons of this approach are that they are difficult to develop broad spectrum antivirals because of its high selectivity. And another important problem is that due to its high specificity, in some cases, it is easy to develop certain degrees of resistance, and the antiviral lose efficacy. On the other hand, host targeting approaches have some pros. They are very appealing for broad spectrum antivirals. They have a higher barrier to resistance because they are directed against a cellular factor, and they are ideal for drug repositioning and turning drugs that target cellular factors into antivirals. This approach is not exempt of some cons. You can the toxicity and side effects because you are considering compounds that are targeted against cellular factors, and in some cases the high dose that is required for antiviral activity impairs its further development, like a real or true antiviral compound. So today I am going to talk to you about host targeting approaches and the part of work that is going on our lab. And why host targeting antiviral approaches? This is the replication cycle of West Nile virus, and if you see the figure, it is intimately ligated to the cellular structure. Specifically, West Nile virus replication is tightly related to endomembrane function, and the virus rearranges these intracellular membranes that mostly came from the ER, and it develops these nice replication complexes you can see here that are called vesicle packets. These are invaginations of the endoplasmic reticulum membrane, and here these are the places for viral replication, genome replication, viral assembly, and particle biogenesis. So you can see from these images that the virus remodels the cell to accommodate its intracellular architecture to its specific requirements. And this is important because for this process, the virus requires cellular factors. And what factors do flavivirus need for this remodeling? Well, first of all are the structural components. I can call them the bricks. And these are specific lipids with a specific topology that enables membrane bending, specific proteins that are involved on different aspects of viral replication, or nucleosides that are important for genome replication and assembly of the infectious progeny genomes. And in addition to these structural components, you also need energy, because all these process consume a lot of energy, and this energy comes in form of ATP. So if we have a look at what takes place in a cell infected with Zika virus, you can see that there is a massive transcriptomic reprogramming that is redirects the metabolism on the biosynthetic pathways to produce the components that the virus prefers. Also, continuing with the example of Zika virus, you can see here that this reprogramming also can be followed and affects the cellular lipids. And here you have some pictures of the Zika virus lipid from infected cells on 24 and 48 hours post-infection, and you can see that there are a lot of lipid species that are altered, upregulated or downregulated, and these species vary with infection time. So what can be extracted from this information is that the flaviviruses modulate the cellular metabolism, and you can see in the graph that the replication compartment meets lipids that came from structures like lipid droplets, which has been very well documented for dengue virus, and also other lipid compounds, and that are fatty acids and it can require fatty acid synthase. But you do not need only lipids or structural components. You need also energy, and a very good source of energy is glucose. Glucose can be transformed to pyruvate and oxidized in the TCA cycle and producing citrate that can be useful, but also ATP by beta oxidation that can also come by beta oxidation of fatty acids. So in our case, we are focusing on this dependence also on glucose for flavivirus replication, and specifically for West Nile virus replication. This can seem something quite novel, but if you go back in the literature, you can find nice studies in 1957 describing the effect of the poliovirus on glycolysis and uptake of glycine by infected cells. And it's more than 70 years ago than these studies were performed. This means that this dependence of virus on cellular metabolism is not a novel issue. So how are we with in this scenario? You can see here that there are a lot of examples of viruses, not only flaviviruses like dengue or Zika, other viruses including hepatitis B virus, human cytomegalovirus, or Epstein-Barr virus, adenoviruses, that require the use of glucose and glycolysis and hijack components from the TCA cycle to promote viral replication. So in a simple view, you can think that this energy and these lipids, these specific lipids, are only important to develop the replication complexes or to assembly novel virions. But this goes far beyond, because the immune response is also related to lipid and energy metabolism. In fact, here you can see that interferon response is tightly associated with adipogenesis or thermogenesis or fatty acid oxidation or oxidative phosphorylation. Another example can be provided by T cells in which the metabolism of glucose and lipid metabolism cooperate to promote a strong source of energy required for the function of these cells. This translates us to the concept of immunometabolism, and it is an important concept that also relates the cellular metabolism with the immune function. And again, this concept came also from more than 100 years ago with the description of the Warburg effect and aerobic glycolysis in cancer cells. Since that, it has been a long way to walk that has identified a lot of aspects of immunometabolism and a specific function of T cells, macrophages, cancer tissues, and things like that. So, what are our targets under study? Having in mind all these things, our targets for these studies related to West Nile virus are lipids and also glucose. This is our interpretation of the bricks, some of the bricks, and some of the components for the energy. And why have we selected these targets? Well, our aim is to identify metabolic changes during West Nile virus infection in vivo, because all the information that I showed you in the introduction, especially that related to Zika virus, has been obtained with cell culture models. And that has some problems, because it depends on the cell that you use. If you use a tumor-derived cell, you cannot be studying properly the metabolic rearrangements, because tumor cells have also their specific rearrangements. So, and this difficults the translation of the results from cell cultures to give candidate therapies. So we have focused on this study to the in vivo aspects of the West Nile virus bioenergetics and metabolic reprogramming in order to try to find new targets for therapeutic approaches. That's you can see in the figure that our idea is to target the viral factory and block the viral factory and hence block the viral multiplication. So let's start by lipids, which has been traditionally our target since almost 1 decade. To study the role of lipids in West Nile virus infection, we use the mouse model of West Nile virus infection. Why use this model? Because West Nile virus is highly neurovirulent in mice. Infected mice develop clinical signs similar to that in humans, including ataxia, encephalitis, acute flaccid paralysis, and the virus exhibits a high lethality. Well, we infected the mice, and you can see the uninfected mice with the circles and the infected mice with the squares, red squares. And you can see that after infection, uninfected mice are happy, they are gaining weight, and infected mice are happy for the first days, but from day 7, they start to lose weight, and you can also record deaths. And this means that the virus is progressing. If we analyze the viral burden in brain, which is the target tissue more important for the neurological symptoms, you can see that from days 7 and 10 post-infection, you have very high titers of virus on this tissue. And this presence of virus correlates with the induction of pro-inflammatory cytokines in brain, like CCL2 or CXCL10. So this means that on days 7 and 10 after infection, you have a neuroinvasion with neuroinflammation, and these are the time points most elective for our experiments, because we think that they resemble the more similar model to neuroinvasive infections in humans. So this is the scheme for the lipid profile analysis in the mouse model. And you can see that we infected the mice with our Neuro99 strain. And you have another control group in parallel that with mice that are non-infected. And we extracted lipids from blood and from plasma from blood at 3, 7, and 10 days for post-infection. These lipids were analyzed by liquid chromatography coupled to mass spectrometry, and in the heat map you can see the lipid profiles changing with time. And at 3, 7, and 10 days post-infection, there is almost no change on uninfected mice. But in the case of West Nile virus infected mice, you can see lipid rearrangement in the plasma of infected mice as early as 3 days post-infection. And these changes vary with the infection time, indicating that these are a dynamic change that is not that varies with the time post-infection. Maybe it's more easy to follow in the box plots. These are the box plots for the significantly changes lipid subclasses that include ceramides, dihydroceramides, dihydrosphingomyelins, phosphatidylcholine, phosphatidylethanolamine, monoacylglycerols, and triacylglycerols. And you can see that there is a tendency or significant that with infective progress, sphingolipids like ceramides and dihydroceramides or dihydrosphingomyelins are increased in infected animals in comparison to uninfected mice. So where do these lipids came from? These are circulating lipids, and you know that the liver is a major regulator of lipid metabolism within host. So we analyzed the transcriptome from liver in West Nile virus infected mice and found a big cluster of differentially expressed genes related to fatty acid metabolism, metabolism of steroids, sphingolipid metabolism, synthesis of phosphatidylcholine, or lipid droplet organization. All these lipids were related to lipid metabolism, indicating that the liver undergoes an important metabolic rewiring during West Nile virus infection. And if we analyze the lipidome of mice infected with West Nile virus and compare it with that of mice uninfected, you can see that there are significant changes in a wide panel of lipid species, lipid subclasses, including sphingolipids that were increased in the plasma of mice. That's suggesting that the liver can act as an important source for these lipids during West Nile virus infection. All this has been performed with mice, but we want to extend this analysis to humans, to infected patients. And thanks to a collaboration with the people from Hospital Virgen del Rocío in Sevilla that suffered from the Spanish outbreaks of West Nile virus, we took access to serum samples from a panel of patients from the outbreak of 2020. And as you can see in the graph, the comparison between patients without West Nile virus disease and patients with West Nile disease, there is a portion of the heat map that is different between these groups of patients, and again corresponds to sphingolipids, ceramide, dihydroceramide, sphingomyelin. You can see this plotted in the box and whisker graph, and you can see that again, patients with West Nile disease exhibit increased levels of ceramide, dihydroceramide, or lactosylceramide in their serum, which resembles that observed for mice. So this put our focus on ceramide, and ceramides are important bioactive lipids with important biological functions, but also with deleterious consequences that are also lipotoxicity that can affect endothelial cells, microglia, or neurons within the CNS. Thus, our approach was to inhibit the production of this ceramide using this compound, the DPTIP-P1 or compound P1, that is an inhibitor of neutral sphingomyelinase 2, that is an enzyme that converts sphingomyelin to ceramide. The experiment is simple. You infect mice with West Nile virus and treat them with DPTIP 1 day before infection and once a day up to 7 days post-infection, and on day 7 post-infection, mice are sacrificed and the tissues are analyzed. Well, if we analyze the viral load in brain, we didn't observe a reduction in the virus load in the brain of mice treated with the compound. This was not similar to what we observed in cell cultures, in which the DPTIP exhibited a very beautiful dose-dependent reduction of infection. But we observed an interesting reduction in the production of IL-1 beta. That is an important immune mediator in West Nile virus neuroinflammation. So these results indicate us that using this class of compounds, we can obtain an immunomodulatory effect on West Nile virus infection, which open new therapeutic opportunities to combat the problems associated with West Nile virus infection. And I am going to move from lipids to sugars and tell you a small snapshot on the work that we have performed with the glucose on West Nile virus. And just for a small remember, glucose metabolism is consists of the oxidation of glucose, usually in conditions that there is enough oxygen available. Glucose is transformed to pyruvate by glycolysis, and this pyruvate enters the mitochondria and is oxidized through the TCA cycle and also produces ATP. In some cases, the oxidation is incomplete and glucose can be transformed to pyruvate and lactate, also in the presence of oxygen. And this is called aerobic glycolysis, which takes place in conditions in which a high demand of energy is required as, for example, as the Warburg effect on cancer cells. So how we study this potential rearrangement in West Nile virus infection? Again, we infect mice and we sacrifice mice at different times of infection, 3, 7, and 10, and comparing infected mice to uninfected mice. We extracted the RNA from the CNS of these mice and performed an RNA-seq analysis, and transcriptomic results indicated performing a gene set enrichment analysis that at 7 days post-infection, the glycolysis was upregulated in the brain of infected mice when compared to its non-infected parallel controls. If we analyze the panel of differentially expressed genes related to glucose metabolism in West Nile virus infected brains, you can see here that West Nile virus infection upregulates the expression of HEK3 and HEK2, the exokinases that are the rate-limiting steps or enzymes for glycolysis, and these are upregulated at 7 and 10 days post-infection. Remember that this was the time that the virus has already invaded the CNS. And we also find the upregulation of a lactate transport at 7 days post-infection. That is important because this is the subproduct of glycolysis. And also we found upregulation of PDK4, pyruvate dehydrogenase kinase 4. That is an enzyme that negatively regulates mitochondrial respiration. This means that an upregulation of this enzyme correlates with a reduction in respiration. And a reduction in respiration promotes an increase in glycolysis. Also, we observed a reduction in some cytochrome components of the oxidative phosphorylation. So this indicates us that the virus is targeting in some way glucose metabolism and specifically upregulating glycolysis in infected CNS. This means with these results, we wanted to try to evaluate whether this can be translated into potential therapies. So we selected 2 reference inhibitors of these enzymes. The first of them is the 2-DG, 2-deoxyglucose, and this is an inhibitor of exokinase. And the other inhibitor is DCA, dichloroacetic acid, that is an inhibitor of the activity of the PDK4. And we tested the effect of these inhibitors again on our infection model of West Nile virus. For this experiment, we use a control group with a drug vehicle that was sodium chloride, and we treated with DCA or with 2-DG. Euthanize the mice at 7 days post-infection, harvest the brain, and analyzes the brains. Well, regarding the viral burden in brain, you can see that 2-DG did not reduce the viral burden in the brain of infected mice. However, DCA was sufficient to reduce this viral burden in some of the animals, and indicating us that the intervention on this pathway, on the glycolytic pathway in vivo, can elicit some kind of antiviral effect for West Nile virus. Moreover, we also analyzed cytokine induction, and we observed that the 2 compounds significantly reduce the expression of different cytokines like TNF-alpha or CCL2. So this reinforced the view that these interventions targeting the energy metabolism in the CNS not only can result in a reduction of viral multiplication, but also have an impact on immune response. You can remember the immune metabolism from the introduction. So this is a clear example of the potential of these therapeutic interventions. And so just to sum the results here presented, we have tried to analyze the effect of West Nile virus infection in the mouse model. And we have found that in the brain, we observe a metabolic reprogramming characterized with an increase in glycolysis. We also have described metabolic rewiring in the liver that also correlated with an lipid rearrangement. And we also have noticed lipid changes in the circulating lipidome of infected mice that are quite similar to the lipid changes in the circulating lipidome of infected patients. So if you are not asleep, I hope that this talk can change your classic view of West Nile virus to my current view of West Nile virus, that is a virus that likes a lot lipids and glucose, and this provides us with important targets that deserve future study for the development of broad-spectrum antiviral therapies. And thank you to all the people in my lab, especially to Patricia Mingo Casas, because a big part of this work formed part of her PhD. And of course, thank you to our founders and also to our collaborators. Thank you for your attention.
Thank you very much, Miguel, for this very nice talk and very interesting, joining the metabolism and also the infection with West Nile virus. And now we have time for questions. So if somebody want to ask something to Miguel Angel, just open the micro and go ahead. Hopefully we don't have problem this time with this. If not, I can start with a question regarding to the Miguel Angel. How specific is your funding regarding to the West Nile? I mean, do you expect change it similar to the one that you described, for instance, in a pathology that is causing an inflammation of the central nervous system without virus, like, for instance, multiple sclerosis, something like this? What do you think about that?
Well, I think some of the findings that we have found resemble some pathology from the CNS, like Alzheimer or inflammation in the CNS. And this is quite interesting because the compound that we are working with, the DPTIP, was initially developed to treat neuroinflammation. And specifically, I think it was developed for treat certain types of brain injury or Alzheimer's disease. So this represents a drug repositioning effort from our part.
Okay, thank you.
We have a question in the chat.
We have a question in the chat, okay. I was wondering if you include box X's in animal model study, and the question is, do you expect similar responses in a mosquito-mediated infection mice model?
Well, these are very good questions. We usually work with only with female mice because of they are more easy to group and to change from cages, and the males we like to fight among them. And for this experiment that are only proof of concepts or for the targets, we prefer to minimize the variability. So, of course, if we find some good therapy, we have to extend it to both sexes. And the other question was that related to the infection with the mosquito. I hope that the results should be similar to those to the mosquito. We do not work with the infection with the mosquito because we have no insectary available at BSL-3 facilities. It is one plan, but we cannot work with infected mosquito in this time. But the patient, the human patients were infected with mosquitoes, and the results, at least in the blood lipidome, were very similar to those from our experimentally infected mice.
Thank you, Miguel. I have another question. I have the curiosity. You focus particularly your result in human sample in those lipids that were increased. But what happened with those lipids that you are observing in the mouse model, a decrease in expression? For instance, the PC that I think that is phosphatidylcholine,
yeah,
and cholesterol and other lipids that you are observing that during the infection with West Nile, they are reducing the expression. So what do you think about this? Because something that is important is you are observing an increase, but at the same time, for other lipids, you are reducing the expression. So what is the balance? What is the final interpretation of this result?
Of course, I agree with you, because we only focus most of time in things that are decreased, in things that are increased, sorry, because medicinal chemists like to develop inhibitors. So it is easy to find a drug that blocks the production of one specific lipid, that a drug that promotes the production of other lipids. And probably this is most just a case of practical issue than other question.
Okay.
We have 4 questions in the chat, so one from you can go, Jorge, if you want.
Okay, yeah.
So
the one question that they say the strain NY99 is known to be highly virulent, and so this might refer to severity of disease, but have you tried with less virulent and maybe more currently fit West Nile virus strain like West Nile virus EG101? Would you expect the same metabolism rewiring and lipid profiles?
Okay, so we are using New York 99 strain because it's highly virulent, and we need that the virus reaches the CNS to reproduce the severe cases of the disease. So we've looked for these strains. We also work with other strains from lineage 2, isolated in our lab, but they are also highly neurovirulent. I don't know if an attenuated strain would induce this rearrangement. In the case of Zika virus, there is a very nice paper that indicates that there are differences on attenuation and pathogenicity based on the strain and the ability of the strain to induce or not glycolysis. So maybe this could be the case of West Nile, but I don't know at this time.
Okay, thank you. We have another question for Mireia Pellegrin. Thank you for this nice and inspiring talk. I cannot use the micro. I have a question regarding the modulation of the immune responses by the glycolysis inhibitor. In addition to cytokine level, did you look at the effect on immune cell recruitment?
That's a very, very nice question. We have not looked at the immune cell recruitment. We only addressed this effect by means of transcriptional rearrangement and bulk RNA sequencing. We have not performed single-cell RNA sequencing or flow cytometry to separate populations. Because just because a practical question in our lab, we have not these techniques setting up, and we are trying to set up the analysis by flow cytometry of the immune population within the CNS, but it's not an easy aspect.
Maybe Miguel Angel, by microscopy, you can also evaluate the presence of the immune cell that are infiltrating.
That's another possibility.
We can go to the next question from Nuria Izquierdo-Ceros. Excellent presentation. Congratulations. What about the possibility of combining drug targeting lipid and sugar at the same time? Would that be possible?
Well, I think it could be possible. I think that a lot of people is affected by metabolic syndrome in humans, and they take statins or something like that, and also beta-glucan or something like that to modulate sugar. So it's a possibility to explore, because we are trying just to dissect the aspects, but I think a combination could be a good idea.
Okay, thank you. We have another question for Federico Bertoglio. Very interesting talk. Cannot use the mic. How close to the clinic is the application of glycolysis inhibitors?
Well, that's a very nice question. One of the glycolysis inhibitors that we have tested, the DCA, is an orphan drug that reached clinical trials for the treatment of a pediatric disease that is called lactic acidosis. So I think it's close to clinics, but probably the pharmacodynamics and pharmacokinetic of these inhibitors that we are working with, including that of 2DG, is not optimal still for clinics. So I think we have identified a good target, but there is still a path to walk for the medicinal chemist to optimize these targets or these candidates and turn them into effective drugs.
Okay, thank you. I have another question from Mar Naranjo. She's interested in is the lipid inhibitor metabolized in the liver impairing its action? Do you plan to use another one taking into account this point?
Well, I don't know where it is metabolized, because this compound we have not designed this compound. It was designed by other group, and we have only used it. And I don't remember where it is metabolized, but I think it could be in the liver, but I don't know.
Okay. We have another question for Cristina Vidal. Do you have any idea of how the virus is triggering this metabolic rewiring? Do you think that the rewiring is made by direct interaction between the virus and the human metabolism, or it should be more by an indirect mechanism?
Well, we have not performed experiments. We would like to address this point in the future, but I think from the evidence that comes from related Flaviviruses, it could be a sum of the two aspects. The infected cells require a lot of energy, a lot of specific lipids, so the cell undergoes a remodelation. And in addition to this, you can see examples, for instance, from dengue virus, that viral proteins recruit specific factors, for instance, fatty acid synthetase, to the replication complex to stimulate the production of fatty acid and the production of specific lipids that they need. So I think these are the both of possibilities.
Okay, thank you. So I think that related with this question, I think that something that is also important to keep in mind with this kind of experiment is also that the immune response at the very beginning of the infection, the immune response, particularly this cytokine like TNF, IL-1 beta, is affecting also to the metabolism by reducing the uptake of nutrient by the animals. So this anorexia stage, initial anorexia state, or that is following to the infection, may also play a role in rewiring the metabolism of the lipid in the liver. In this, and this is something that is physiological. I mean, from the immune system. So, do you think that it's possible to discriminate, to separate what is the effect of the virus and what is the effect of the immune system, or is a complex process in which both are involved and it's not going to be possible to discriminate between what is the key element that is governing these changes?
I think that you are totally right. The metabolic rearrangement that we observe have to be a sum of the infected cell and a sum of the metabolic rearrangement. We have tried to discriminate the metabolic rearrangement in culture cells because we use cells, virus cells that have no immune system. And when we infect virus cells and use the real-time metabolic analyzer, this is called seahorse. There's a you infect the cells and put it on the seahorse, and you see an upregulation of glycolysis only for viral replication. Even more, this can be resembled not with infectious virus, but also with a replicon. With a subgenomic replicon that is only replicating, it is enough to upregulate glycolysis. So we can assume that in infected cells, there is also an upregulation of glycolysis, but also in infected tissues, there is a homing for cells from the immune system, and also the cells change their metabolism. So for us, it is important to analyze the whole process, because the compound that you give to the animal or the drug is also going to go to infected and non-infected cells as well as to immune cells. So you have to think that the expected outcome comes from the sum of all the players.
Okay. And regarding to I have another question where we can wait for more questions for the audience, but in the middle I have another question that is regarding something that is very, very nice is the result that you are showing, the timing of the changes that you are observing. Do you think that this reprogramming in the metabolism is playing a role in neuroinvasion, or is more related with the particularly with the neuroinvasion? I mean, is a step previous to the neuroinvasion that the metabolism is changing, and that make that the virus can reach the central nervous system more easily, or is a consequence of this neuroinvasion?
We don't know. Maybe the suggestion of using strains that are not so neuroinvasive can help us to ask this question. If we observe the early arrangements, it's clear that it's the virus and not the neuroinvasion.
Because with the inhibitor for the glycolysis, you observe a reduction in the virus, in the viral load, at least you show a trend in the reduction in the virus, but also you are observing a reduction in the cytokine that you are measuring, the CCL2 and also the TNF in the brain. So maybe this inhibitor is in some way modulating or impairing the access of the virus to the nervous system.
Yeah, maybe it is impeding the access, or it is reducing the neuroinvasion, both things. Yes.
Okay.
And it's also there is a very nice paper written from another group that has also analyzed glycolysis in West Nile virus infection, and they have found that macrophages are highly affected by 2DG. This is quite recent, 2 months ago or something like that. So this reinforces the idea that the neuromodulation of the neuroinflammation can be performed with this kind of inhibitors. And indeed, it is also important because the damage caused by West Nile virus does not only came from neuronal death, it also came from neuroinflammation, it's also an immunopathology. So maybe we are focused on reducing viral replication, but we have also to reduce viral replication together with neuroinflammation.
Yeah. We have another question from Mira Tabadia. have you been able to identify the specific ceramide and sphingomyelin species? Does carbon length play a role in the infection? Also, is the novel pathway involved, or is ceramide mostly produced through hydrolysis of sphingomyelin?
Okay, so
you have here 3 questions.
Yeah. It's 3 difficult questions. Well, we have identified the length of the ceramides. Indeed, in the lipidomide that I presented, I think we are moving around 10 to 15 species of ceramide or dihydroceramide or this kind. We have not observed a direct relation between carbon length and increase in specific ceramides. We observed some specific species changing, but we have not find a correlation. And regarding the novel pathway, this is a very beautiful question because we have focused on the production of ceramide from sphingomyelin to ceramide because we have very good results from in vitro experiments for more than a decade ago. We identified the neutral sphingomyelinase as an important enzyme for West Nile virus replication, and this enzyme has been also involved in Zika virus replication. So we are interested on this pathway, but we have also performed experiments in mice that have higher amounts of ceramide than wild-type mice using a model of infection of a metabolic disease. It is called Niemann-Pick disease type A. That has a mutation in the acid sphingomyelinase, and the mice accumulate high amounts of sphingomyelin in the tissues. And these mice are highly susceptible to West Nile virus infection. They die before wild-type mice. So the question is beautiful because sphingolipid metabolism is quite complex, and we are observing that at least this branch of sphingolipid metabolism can reduce viral multiplication, but on the other hand, accumulation of sphingomyelin can promote viral replication. So the axis ceramide sphingomyelin, it's quite interesting for West Nile virus infection.
Okay, thank you. More question? Okay, so if we don't have more questions, I think that now is on time. It's almost 4 p.m., so we can end here. I would like to thank again Miguel Angel for joining this seminar and very interesting talk. We invite you to follow this seminar by the next month. Hopefully we can arrange another talk. So thank you very much to all of you for joining this meeting, and enjoy the rest of the day. Bye-bye.
Thank you to all the audience, and especially to you, Jorge, for this kind invitation.
Thank you. Bye-bye.